Hello there, guys. Well done for logging on and turning up for the last lesson for lockdown learning. Congratulations to getting this far through the term and for sticking with us. Today's lesson is going to be a little bit different from the other lessons that we've done previously. So today's lesson, we're going to basically be going through the answers of a test that we've uploaded onto class charts. So if you guys haven't had a chance yet to find that test or to do that test, stop the video now, go over to class charts. You can either print off the test and complete that, keep completing on that, or you can use a notebook or a piece of paper and just record your answers from that test onto that. To do the test, you'll need a pen or a pencil, paper or notebook, or as I say, a printed copy from the exam that you've got from class charts, and you will need a calculator as well. Okay, so they're the three main things you're going to need to be able to do the test. It's been uploaded onto class charts, so if you haven't done so already, stop the video, go find the test and have a go through the test. It's not a very long one, guys. I think I put 13 questions on there for you to have a go at. Give yourselves about an hour to do it, a little bit longer if you need it, shorter if you don't. But as I say, beyond this point, we're going to be going through the answers. So if you haven't done the test now, stop the video at this point, go away, do the test, come back after you've had a go at the test, and we'll be able to go through the answers from there. Okay, guys, well done. Hopefully everyone who's at this stage of the video has had a go at the test. I hope you didn't find it too difficult. Most of the questions there were stuff that we've covered during our lockdown lessons. And so what I'm going to do is I'm just going to crack straight on, guys, and we're going to start we're going to move straight on to the questions in the exam. So our first question was, yeasts are eukaryotic microorganisms that are used in the brewing and baking industries. The diagram shows a yeast cell. State two ways in which the structure of this yeast cell differs from the structure of a bacterial cell. Now, if you can remember, bacterial cells, all bacterial cells are prokaryotic cells, whereas yeast cells, as we can see in the question, are eukaryotic cells. So that's what the question's basically asking us, to see if we can determine the differences in structure between a prokaryotic and a eukaryotic cell. So my answer was that the DNA of the yeast cell is held in the nucleus, whereas the DNA of a bacterial cell is loosely coiled in the cytoplasm of the cell. Now, that's the key distinction between a prokaryotic and a eukaryotic cell. Prokaryotic cells have no nucleus, eukaryotic cells have a nucleus. So that's the key distinction. So hopefully you all got that one. The next one, actually, you could have used, for the next statement, the next fact, you could have used a range of answers. I've gone for many bacterial cells have a flagellum for movement. Yeast cells do not have this organelle. So that's another difference. You could have used slime coat. You could have used plasmid DNA. There's a whole range of different organelles that the prokaryotic cells have that the eukaryotic cells don't have. You could also say that a eukaryotic cell has mitochondria, whereas a prokaryotic cell doesn't have those. So whichever ones you've gone for, guys, give yourself a mark for each one of the structural differences you've selected. As I say, I hope everyone went for the nucleus. The eukaryotic cell has a nucleus, and as I say, you're just looking at one of the other differences. So eukaryotic cells have mitochondria, prokaryotic cells don't. That's one you can give yourself a mark for if you put that down. Also, flagellum, a bacterial cell has a flagellum. They also have a slime coat and a plasmid or a plasmid. And so if you've selected any of those three organelles saying that the prokaryotic cell has them and the eukaryotic cell doesn't have them, you can give yourself another mark. So that's 2 marks for that question, guys. Hopefully we're doing all right. So I'm going to move on to the next question. What is the function of a ribosome? Now, it's only 1 mark on this, guys. You only need a very short answer. And basically, it's the production of proteins from amino acids. Your ribosomes are the part of your cell that forms all the proteins by combining amino acids. So, question number 3. Figure 7 shows some Vibrio cholera, the bacteria that causes cholera. Now, in this image, the length of one of the flagellum on figure 7 is 68 millimeters. The question is asking you to calculate the length of the flagellum in micrometers. Now, we do have all the information that we need with the image. If you look down on the image, we can see how much magnification, how big the image has been magnified. And we know how big it is on that image, 68 millimeters, and it's been magnified 8,000 times. I believe in the test I did give you the equation that you would need to do, but if I can't remember now, but that's the equation you wanted to be using. Actual size is equal to the image size divided by the magnification. So as long as you've got that right, you'll hopefully be able to just input the numbers and then come up with somewhere near a correct answer. So the actual size is 68 millimeters divided by 8,000. That's the number of times of magnification. And that gives us an actual size of 0.0085 millimeters. Now, if you've put that as your answer and you've included millimeters, you have to have included millimeters, you can give yourself 1 mark. And the reason you can only give yourself 1 mark is because the question asks us to calculate the length of flagellum in micrometers. So there's another stage we need to do this. We need to convert our millimeters into micrometers. And to do that, a micrometer is 1,000th of a millimeter. So to convert our size from millimeters into micrometers, we need to multiply by 1,000. So we do that. And if you have put in 8.5 micrometers, that's what you needed to put down to be able to acquire full marks for this question. If you've written 0.0085 millimeters as your answer, give yourself 1 mark. If you've written 85, I'm going to be really kind, guys. You can get 8.5. You can give yourself 1 mark. The only way that you can get full marks for this question, though, is if you have got this number, 8.5, and micrometers as your units. The units are very, very key, guys. Okay, so give yourself a couple of marks, or give yourself your marks for what you've got on that question. And we'll move on to the next question. Okay, the diagram represents an enzyme catalyzed reaction. What is the name of each labeled part? So this was our enzyme lesson that we did. We had a couple of lessons kind of covering this topic. So A is the enzyme. We can tell that because we can see this intricate shape here, and that would be the active site. B is the substrate. That's what's going to fit into the active site. C is the enzyme substrate complex. That's what they're called when the two are the enzyme and the substrate are combined. And then D is the products of the reaction. That's what has been catalyzed by the enzyme reaction. Okay, so A enzyme, B substrate, C enzyme substrate complex, and D products. Give yourself a mark for each one of those that you got right, guys. All right, we're going to move on to this next question. Explain the effect of temperature on the activity of lipase as shown in the graph. Now, it's worth 3 marks, this question, guys. So when I look at that graph, I can quite clearly see 3 clear distinctive parts to that graph. So when I would try, if I was answering this question, I would try and have a comment about this upward trend section of the graph here. I would have a comment about the upper section of the graph, the top part up here, and then I would have another comment mentioning what the downward trend is representing here. And it's to explain the effect of temperature on the activity of lipase as shown in the graph. That's the question. So we're going to explain what the changing temperature does to the activity. So the first part of that question is as temperature increases between 20 degrees Celsius and 36 degrees Celsius, the activity of lipase also increases. This is due to an increase in the chance of the substrate and enzyme colliding, coming together. So that would give me 1 mark. That's the first of the points that I would be looking for. If you've said something along those lines, obviously give yourself a mark. Now, I would like the second statement that I would add for my second mark for this question is that at approximately 37 degrees Celsius, lipase activity is fastest. This is the optimum temperature for lipase. Now, the key statement in this is optimum temperature. I really do hope that you guys are using the appropriate scientific terminology whenever you're completing these answers. But that's the statement I would do there. That was, so far I've got 2 marks for this question. And then the third statement is that as the temperature increases beyond 37 degrees Celsius, Lipase begins to become denatured. As the enzyme is denatured, it changes shape and the substrate can no longer bind with the active site. This rapidly reduces the rate of activity of lipase. Now, as I say, guys, you don't need to have put that word perfect, obviously, to have got the mark, but as long as you've mentioned that during the increase of temperature on this side, the rate of reaction increases. At this point here, at the top of the graph, that's the optimum temperature for lipase to be functioning, and that due to denaturing of the lipase protein, that actually the rate of reaction falls off very quickly because the enzyme can no longer bind with the substrate. So there's 3 marks there. Words to these effects, guys. Give yourself a mark out of 3 and see how well you've done on that question. Okay, so the next question, it's a 1-mark question. Osmosis is a method that single-celled organisms, such as bacteria, use to obtain molecules from their environment. It's a little bit misleading, actually. It should say water molecules, because that's what osmosis is transporting. Which of the following is a correct description of the process that involving the transport of molecules? So this is our diffusion, osmosis, and active transport section of the lockdown learning that we did. And so we go through the answer. We've got a multiple choice. Diffusion is used to transport molecules against the concentration gradient. Well, we know that's not true because diffusion is a passive mode of transport, and it goes down the concentration gradient. So it doesn't go against it, it goes down. It goes from a high concentration to a low concentration. So we know A isn't right. B, active transport is used to obtain molecules in a low concentration environment. Now there's nothing about that stands out as being incorrect. To my mind, that sounds like that might be correct. So we'll have a look at the other answers to make sure that there's nothing else that's more suitable. C is active transport moves substances along the concentration gradient. Again, we know that's not true. We know that active transport moves substances against the concentration gradient, and that's the reason it requires energy to be able to do that. So we know that C is not correct. And D is diffusion uses energy to transport molecules into cell. And again, I said at the top of this question, diffusion is a passive mode of transport, and as such, it requires no energy. So we know D is not right, and that means that we were quite right in our surmising early on. B is the correct answer. Active transport is used to obtain molecules in a low concentration environment. Give yourselves a tick if you got that one right, guys, and we'll move on to the next question. Okay, osmosis and diffusion are processes used in nature to transport substances into and out of cells. State 2 ways in which osmosis and diffusion are similar. Now, the ones I've gone for, guys, is that both osmosis and diffusion are passive forms of transport. They require no energy. Okay, so that's fact 1. That gives me my first mark. If you've said something along those lines, give yourself a mark. Osmosis and diffusion both transport substances down a concentration gradient. In both cases, they both go from a high concentration down to a low concentration. So if you've said that fact as well, you can give yourself a mark. You can also give yourself a mark if you said both occur in nature, both involve the transport of particles or molecules. These are 2 acceptable answers that you could have put outside of the ones that I've put on the board in front of you. Now, the next question was state 2 ways in which osmosis and diffusion are different. Now, there's less answers that you could have given, actually, for this question. The first of them is diffusion involves the transport of solutes, while osmosis involves the transport of water. And that's quite a key distinction. So with osmosis, it's always water that's traveling down the concentration gradient, whereas in diffusion, it's the stuff that's not water that's traveling down the concentration gradient. It's the solutes. And a semi-permeable membrane is essential for osmosis to occur. Diffusion can occur across a membrane, but it is not essential. And that's a real key distinction, guys. So hopefully you've been able to come up with those 2 key ways that osmosis and diffusion are different. And as I said, we've got the ways that they were similar and the other answers that I alluded to as well. So give yourselves a mark, 2 for each one of those questions, and we'll move on to the next question. Okay, the diagram shows the chambers of the heart. Explain why the wall of chamber B is thicker than the wall of chamber A. Okay, so we're looking to discuss the left ventricle having a thicker wall. Now, if you can remember back to our heart lesson, the reason it has that thicker wall. Let's put the answer up and we can go through the answer, shall we? Okay, so chamber B has to a left ventricle has to pump blood around the whole body. Blood that's leaving chamber A, the right ventricle, only passes through the lungs before it was returned to the heart. To be able to pump the blood around the whole body, chamber B must pump the blood at much greater pressure than chamber A. To create the greater pressure, chamber B has a thicker muscular wall. Now, there's a lot of points in that answer there, guys. Basically, what I would like somewhere in your answer to include is that chamber B is pumping the blood around the whole body, and in order to do that, whereas chamber A is only pumping to the lungs and back, and in order to pump the blood around the whole body, it needs a greater pressure, and that is the reason why it has those much thicker muscular walls. So as long as you've got in that area your answer, have a look, read through it, and give yourself a mark, either a 2 if you've got both two key facts, the fact that chamber B is pumping it around the whole body and it needs more pressure, and that's why it has the thicker walls. They're your 2 key points that you're trying to get out of this answer. So we're going to move on to the next question, guys. Which of the following shows the direction that blood flows towards, through, and from the heart? So again, it's a multiple choice question, guys. We can go through this. Whenever I get these kind of questions, I always have a go for one because it's multiple choice. And even if you've got no idea, you've got a 25% chance of getting the question right. So always have a go at multiple choice, guys. But I'm going to go through these answers now. And it's just a case of process of elimination with these kind of questions. So the first question says the blood flows from the vena cava. Now, there's no problem with that, into the heart. The problem I have is that it's saying that it flows straight into a ventricle. Now, I don't know if you can remember, but the vena cava flows into the right atrium first, and not the right ventricle. It's the ventricle that pumps the bloods out of the heart. The atrium is where the blood is received before going into the ventricle. So we know that the vena cava does not deposit blood in the ventricle. So we know that A cannot be right. Pulmonary artery into the atrium, into ventricle, into aorta. Now, again, we've got a bit of a problem here. The pulmonary artery, the word artery begins with the letter A, and therefore we should know that the arteries carry blood away from the heart. So if the blood's in the pulmonary artery, its next part of its journey is not going to be the atrium. If it's in the pulmonary artery, it will be going to the lungs, then the pulmonary vein, before returning to the atrium. So we know that this one's not correct as well. C, the aorta, is the main artery that takes blood away from the heart. And we know, therefore, that cannot be the correct answer. The aorta does not return blood to the ventricle. So the last one we've got is the pulmonary vein, returning oxygenated blood from the lungs to the heart, enters in this instance, it would be the left atrium, then into the left ventricle, before it is then pumped into the aorta and around the body. So through that process of elimination, guys, we were able to eliminate A, eliminate B, eliminate C, and that gives us D as our correct answer. Give yourself a mark if you got that one right, guys. And we'll move on to the next question. Okay, more carbon dioxide is produced by cells during exercise. As we exercise, our cells need more aerobic respiration, and they produce more carbon dioxide. Name the part of blood that transports most of the carbon dioxide to the lungs. And the answer is quite simply, guys, it's the plasma. That carbon dioxide dissolves into that liquid straw-like fluid that suspends all the cells and carries waste products and nutrients around in it. And it's that component of the blood that transports our carbon dioxide to our lungs. What vital function do erythrocytes, red blood cells, perform? Well, hopefully you guys are all going to remember this is one of the key functions of blood, and that is to carry oxygen. It picks oxygen up from our lungs and it carries it to every cell in our bodies. state two ways that erythrocytes are adapted for this function. So this is the specialization of red blood cells. They're highly specialized for the job they do. And I've put down 3 facts in my answer. You could have put down any 2 of these 3 facts to give yourself full marks. But the first one is that erythrocytes have no nucleus, and the next one is that they have a biconcave shape. And both of those facts are combined to enable the red blood cell to have a very large surface area to improve their rate of diffusion. The other fact you could also have included is that erythrocytes contain a protein called hemoglobin, and it's that protein that they use to grab hold of the oxygen and to store the oxygen as it's being transported around the body within the red blood cell. So any one of those 3 will give you any 2 of those 3 statements will give you full marks for that question, and then we need to have put down that they carry oxygen for the first part of that question. So moving on to the next question. Okay, so we're nearly there, guys. It won't take long now. An elite athlete has a stroke volume of 100 cubic centimeters. When not exercising, their heart rate is 50 beats per minute. When they're exercising, their heart rate increases to 110 beats per minute. Using the formula triangle, calculate their cardiac output. Okay, so we've got the formula triangle there. Now, I don't know if you can remember back from the lesson we did, it's only a few weeks ago now, but if we're using a formula triangle, what we have to do is cover up what we're trying to find out, and that shows us what the rest of the sum should be for us to input to be able to calculate the right answer. So we're looking for the cardiac output, and hopefully, guys, you've all put down the same equation there. And if you've put down the same equation, then it's just simply a case of copying the numbers that you've got up there and doing the right maths. So heart rate's 50 beats per minute. You times that by the stroke volume at 100 cubic centimeters, and that gives you a cardiac output of 5,000 centimeters cubed per minute. Now, again, I'm going to bang on about this, and I will do all through next year as well, guys, and years to come, I don't doubt. You have to have put the unit down. I'm being really lenient on these questions at the moment. I've given an option of 2 marks that you can possibly score. If you put 5,000 down, you can give yourself 1 mark. Going forward, I don't know that I'll always be this lenient, but if you put 5,000 down, you can have 1 mark. And then you need to have put down an appropriate unit. It could be milliliters per minute. It can be centimeters cubed per minute. You could have written the word per between centimeters cubed a minute. You can do this -1 symbol to resemble the same fact that it means centimeters cubed per minute, or you could have put a /. Don't mind how you've arranged your unit or how you've put your unit up there, but you must have put the appropriate unit in to score full marks for this question. Now, the second part of this question, again, we're looking at exactly the same equation. We're looking at cardiac output, the heart rate times the stroke volume. So the heart rate's 110 when they're exercising, and the stroke rate's the same. So that gives us cardiac output of 11,000 cubic centimeters per minute. Now, again, you can give yourself 1 mark if you've got the right number, but to get full marks for this question, you will need to have put the unit for both of those questions. But well done for having a go at that, guys. We're going to move on to the last question. Okay, compare and contrast aerobic and anaerobic respiration. Now, this is quite a common 6-mark question, and you need to know what you're going to be doing with this question. So compare and contrast, you're looking for similarities and differences between aerobic and anaerobic respiration. So my answer, I've got a model answer here, says both aerobic and anaerobic respiration are exothermic reactions that involve the breakdown of glucose to release energy. So we've got a couple of similarities in there. Aerobic respiration occurs in the presence of oxygen, but anaerobic respiration occurs without oxygen. So there's a contrasting fact there between the two. This can be shown in the equation for aerobic respiration, which is glucose plus oxygen. It goes to carbon dioxide plus water and energy, as we know. The presence of oxygen allows for the complete breakdown of glucose into carbon dioxide and water. Anaerobic respiration can be shown as glucose goes to lactic acid. The lack of oxygen in the anaerobic respiration results in the incomplete breakdown of glucose to lactic acid, and this releases far less energy. So that's my complete mark. That's given me, I would say, 6 marks. We can break it down a little bit though, guys. So if we just pop to the next slide, we can see that that last answer contained all of these facts. So I changed it around. I moved them around them so that the paragraph structure made sense and it all flowed through a little bit easier. But with regards to marking your answer, if you've mentioned aerobic respiration occurs in the presence of oxygen, but anaerobic respiration doesn't or occurs without oxygen, that's definitely a mark you can include there. If you've said that aerobic respiration is more efficient or produces more energy, or you've actually used the term 38 ATP compared to anaerobic respiration, which is only producing a small amount of energy or much smaller, less energy, and if you've used the numbers or whether you've, as long as you've made that distinction, there's another mark you can have there. You can mention the fact that aerobic respiration produces water and carbon dioxide, whereas anaerobic respiration produces lactic acid. You can also mention that both reactions are exothermic. They both produce energy, so they're both considered exothermic reaction. And they both involve the breakdown of glucose. So glucose is the one of the reactants or the main reactant for both of those reactions. And you can also, if you've also, so there's a mark there for that, and there's also marks if you've included the formula, if you've correctly put the appropriate word equation into your answer in the way that I did. There's also a mark there. You can have a mark for both of those equations. So if you put the 2 equations in, that's 2 marks, and any one of those other points that are on the board there, if you've made a mark along a comment along those lines, then give yourself a mark out of that. This whole question will be a mark out of 6. There's several marks available on the screen, but you only need to be at a maximum mark of 6. So mark your work, guys. Give yourself a mark out of 6 of that, and other than that, it's really well done. You've got to the end of lockdown learning, guys. This is our last lesson before we break up for summer holidays. So what you can do at this point is you can add up your marks. The test was out of 34 total marks, so if you add up your marks, you can then, if you want to, you can calculate what you got as a percentage. And to do that, you take your score, you then divide your score by 34. Whatever that answer is, you then times that by 100 and you would get your percentage. For example, if you had, say, 17 marks out of the possible 34, you would do 17 divided by 34. That would give you 0.5. You then times that number by 100, and that would give you 50%. So that's how you can work out your percentage. And at that point, guys, I'm going to say thank you ever so much for engaging with the lessons that we put on. I've been chatting to teachers from a number of schools, and actually you guys have done really, really well in comparison to some of these schools, other schools in the area. You've really engaged really well with a lot of the lessons that have been put up. Hopefully you didn't find the test too hard, but really all that's left for you guys to do now is to relax and enjoy your summer holidays. Have fun, enjoy the lovely sunshine that I think we're going to have throughout this summer holidays. Please be aware that we are still in the middle of a lethal viral pandemic. So guys, keep yourself safe, observe social distancing, wash your hands for 20 seconds as regularly as you can, and just keep yourself safe, because I look forward to seeing you all in September. Cool. Take care, guys, and have an amazing summer holidays. Well done. Congratulations. I'm trying to find stop video.